field of microbiology didn't start until relatively recently, probably 200 to 300 years ago, because of the fact that the organisms that we're studying, while present here around you as you sit in this course, are not immediately apparent. Their existence is not immediately apparent. So think back when you're reading the first chapter of for homework this week about how it used to be before we knew that microorganisms existed. Okay, there were still people who got sick. There were still physicians who tried to treat those people and nurses who cared for them. But the very essence of what was causing their symptoms and illness was not known. Okay? So these organisms that we're studying now, we know are microorganisms. So what really is a microorganism? Or, as I'll often say, a microbe, which is more kind of an umbrella term. It includes microorganisms and microbial agents. Okay? Okay, and so this slide gives an overview of the entire community or group of microbes that includes both microorganisms and microbial agents that are members of the microbial world. Okay, so this is that invisible world that I aim to introduce you to this semester, and that you'll really be delving into and studying the characteristics of not just the anatomy, but the physiology and the pathogenesis of the ones that are associated with us as human hosts in causing infectious diseases. Yes, question? Yes. So again, it'll be a slide set like I showed you for chapter 1. Yep. So just like biologists like to classify organisms into different groups, microbiologists do too. And so all microbes that are microorganisms fall into one of 3 domains of life. Okay, so these are the taxonomic domains that you may have learned about in general biology: bacteria, archaea, and eukarya. And the difference, the main difference is in the anatomy between these organisms. Prokaryotes, cell structure being unicellular, having a very simple organization or anatomy of no internal compartments. Okay, no nucleus, no mitochondria, no Golgi apparatus or endoplasmic reticulum. Okay, they still carry out the necessary metabolism of living organisms. Okay, but they do that without those internal compartments in organization. They tend to be very small as such. Megan? Yeah, basically. Yes, there is a region that we call the nucleoid, but it's not separated by a membrane as it would be in eukaryotes. Okay? So bacteria and archaea fall into those categories. Bacteria, we're all familiar with. Those are the germs that cause disease for us. They're the most common human pathogen. But archaea you may not have heard of. They're often referred to as extremophiles, but there are actually archaea. The more we learn about the human microbiome through a project called the Human Microbiome Project, which is similar to the Human Genome Project, in that it aims to kind of identify what is normally associated either on or in our bodies as far as microorganisms and microbial agents. we find that actually there are some archaea that we can find associated with our body. But they don't cause disease. None of them that we have discovered yet cause disease. And they're often found in very extreme environments, like the geysers of Yellowstone Park, or salt lakes, like the Dead Sea. And in very cold, warm, high pressure, high salt, physiologically uninhabitable, what we would think of uninhabitable areas. But then bacteria are very ubiquitous as well. They're pretty much in any environment you would find. So both very adaptable, very very diverse in their capabilities metabolically. If there's no carbon source there, they can still live and as lithotrophs and live off rocks and inorganic materials. Okay? And then the eukarya is a domain that includes us as humans. But as far as microorganisms, it includes algae, protozoa, fungi, and helminths, which are technically not microorganisms because you can see the parasitic worms of helminths without a microscope. However, we do lump them in with studying microbiology because they are human. Some of them are human pathogens. And they have stages of their life cycle that are very, very small and invisible, small, unable to be seen without some kind of tool to aid in magnifying them. And then we have, beyond the domains of life, we have the non-living or microbial agents, which include viruses, which are also a common human pathogen, but include plant viruses, which are called viroids, and infectious agents that have what is actually mind-boggling to biologists, but no genetic material. However, they cause degenerative neurological diseases like mad cow disease, scrapie in domesticated animals like sheep, and Creutzfeldt-Jakob syndrome in humans. Okay? So we'll, just as an introduction to these members of the microbial world, as I mentioned, they have a very diverse or distinct anatomy, the microorganisms. The ones that are in the domain eukarya have a true internal membrane bound, so that lipid bilayer around their internal compartments. That includes the biggest, or the main one, the nucleus that carries the DNA, as well as other organelles like mitochondria. Yes, Megan? It's thought that probably, as far as like a cellular organism, probably bacteria are the oldest and the pre-existing, and they might have had just RNA type of organisms before that, similar to kind of virus that didn't really have a cellular structure but had genetic material. So that's the current evolutionary biology hypothesis. So we'll learn a little bit more when we talk about the diversity and the classification of how they're related, how it's possible that by symbiosis, by one early ancestor of a eukaryotic cell engulfing another organism like a bacterium, that it became what we know as today as a mitochondrion, that organelle within the eukaryotic cells, all eukaryotic cells. All right. And eukarya are more complex in their cell structure, but they also can be multicellular in their organization, such as helminths. Okay? Going back to the prokaryotic cell structure, the domain bacteria are characterized by a one-cell unicellular structure. They have a wide diversity because they exist in many different environments, from being pulled out of ice samples from the Antarctic to down in deep sea vents in the bottom of the ocean floor. But they, as we'll find, what's useful in for medical purposes is to use these, their outward appearance, so these morphology of how they look under the microscope, to distinguish them as kind of a first step in distinguishing them, and to use staining like gram staining, which you'll read about in Chapter 3, and we'll be doing in the lab, to quickly tell the difference between the two major groups of pathogenic bacteria, the gram-positive bacteria and the gram-negative bacteria. And the shape is determined by the fact that they have a cell wall. So we as humans, our cells do not have a cell wall, they have a cell membrane. But in addition to a cell membrane, bacteria have another layer outside of that cell membrane that we call the cell wall. They often have additional layers, protective layers like capsules, which we'll learn more about in Chapter 5 in more detail. But you should just know for now that it's unicellular, prokaryotic in its cell structure, and that it has a rigid cell wall that determines the shape. As unicellular organisms, they are asexual and have an asexual form of cell division in which one cell basically makes a clone of itself by dividing, fission. So binary 1 becomes by 2, one cell to 2. And because they have a very rapid growth cycle, 1 to 2 and 2 to 4 can result in, and the rapid growth cycle can be as little as 20 minutes, can result in millions of bacteria, as we will see in our experiments. With just an overnight incubation with food, we can see plenty of bacteria growing on our plates or in our liquid cultures. So rather than the diversity that we see with us as sexually reproducing creatures, where our children are very distinct from us in our appearance, these are clonal populations, genetically identical to their parent cell. But they do have ways of sharing genetic information. We'll get to that later in the semester. And they can be, not all, but some can move around and have appendages like flagella that help them to move to areas of food or away from areas where there are toxins like antibiotics. So it's actually a virulence factor to have that trait of motility. One key difference between some pathogens that are bacteria and others that are not. We'll be talking about the metabolism, very diverse, not just chemotrophs like us, but bacteria can use light, they can use organic material or inorganic material to live on. As I mentioned, they're only unicellular, but they can form communities, which are particularly challenging to treat medically, called biofilms, where you have a mix of bacteria growing together in one place. That makes it hard for our immune system to clear it and hard for the drugs that we try to treat these infections with to clear it. And the cell wall is important for identification. It's distinct from other microorganisms that, like fungi, that could be pathogenic, that have a cell wall, but a different chemical structure. And so it can help us to distinguish a bacterial infection from fungal infection. And they acquire foods by simply either having a transport channel across that plasma membrane that divides them from the outside environment, or by diffusion, either active or passive transport. Very diverse characteristics, so the shapes help us to distinguish them quickly under the microscope. And indeed, they are a major human pathogen, many of them. Of course, I say many of them are human pathogens that we really understand, but that is only a fraction of all the bacteria we know. Bacteria are everywhere, and so probably 1% of bacteria or less are associated with the human body, and another less than 0.1% are actually human pathogens. So a tiny, tiny fraction of the bacteria that exist in our world are what we're going to really look at in detail this semester. Archaea, we're not going to spend too much time on, but I want you to know the basic features that differentiate it from other members of the other microorganisms in the microbial world. Like bacteria, they are unicellular and have a prokaryotic cell structure. They have similar shapes because they also have a cell wall that defines their shape, but their cell wall is not peptidoglycan. It's different chemistry, so it can be distinguished by the chemistry of the cell wall, as well as usually by where you find the archaea. So they tend to have pseudopeptidoglycan or pseudomurine or other diverse chemicals as their in the structure of their cell wall. And they're found in extreme environments, very high temperatures, very low temperatures, very salty conditions that would other bacteria and other microorganisms wouldn't be able to survive in. They also have a very diverse metabolism using both organic and non-organic sources of carbon. It's not clear because they haven't been studied that much whether they can form biofilms, but they are all unicellular, and they have a similar means of reproduction, but they're not human pathogens. Okay. In the domain eukarya, the three major groups that we'll be studying are algae, fungi, and protozoa. We'll touch upon helminths a little bit, but you should know that there are some fundamental differences between these organisms that make it easy to distinguish them. While they all share the same cell structure, only some of them have a cell wall, algae being an example of that. And algae are photosynthetic. So unlike fungi, which are not photosynthetic, but some bacteria may be photosynthetic, it's really a key characteristic for they are limited to areas where light, so usually found in the water, your seaweed and your things like diatoms and algae, sometimes found on land, but at different levels of the water because they're all photosynthetic and contain chlorophylls that help them to use that light energy in their producing energy for their metabolism. And they're the bottom of the food chain, like plants, where they're autotrophs, where they produce food as part of their metabolism. They have a cell wall. It's different from bacteria in that it's made of cellulose, so more similar to plant cell wall. And they have chloroplasts similar to plants. They are not human pathogens. However, they can, some of them, cause human disease indirectly by the production of toxins in shellfish, for example, which then, if we consume the shellfish, can make us very sick. Fungi are, again, eukaryotes, and they also have cell wall, that their cell wall is composed of chitin, a variation on the glycan structure that's found, that's peptidoglycan, so it's still glycans, but it's a different chemical organization of those glycans that leads to the chitin. Chitin you may have heard of is in like beetle shells and things like that. They are all organotrophs, so they rely on organic sources of carbons, and they are all heterotrophs, so they need some outside source of nutrients, which they absorb. Some of them are unicellular, some of them are multicellular. You know, fungi are mushrooms, but they're also yeasts and part of our normal microbiota. But they can be pathogens, usually opportunistic. So you usually don't get a fungal infection unless you've either had, by accident, the fungi has been introduced to your body directly, or you have really high exposure, like to the spores of mold you can get sick from, or if you're somehow immunocompromised. So like there was, I think 2014, when people were going in to their doctors to get treatments with cortisone, and they were getting a shot, and the cortisone had a fungus in it that's not a known pathogen, but caused meningitis in many of these people because it was getting across the barrier we have of our skin directly into our bloodstream and spreading and growing in our body. So opportunistic, you know, if it's there and in that nutritious environment, then it can use us as humans as a host to grow. Protozoa are kind of the leftovers of what biologists couldn't quite find a group to categorize in. They're a very diverse group, but they are unified in that they are all unicellular and they all lack a cell wall. So these are protists that have those two common characteristics, but they are quite diverse, from an amoeba that's just a unicellular organism that has a simple life cycle to things like Plasmodium malaria, which causes malaria, that has a very complex life cycle in different hosts. And one characteristic of that's also common to protozoa is that many of them are able to move either by pseudopods, cilia, or flagella, or by secretion of enzymes that help them to kind of glide over their habitat. They are all, again, chemoheterotrophs in their metabolism. Again, all unicellular. They don't have a cell wall, which is a key distinction, and some of them are human pathogens, malaria being one of the leading causes of death worldwide. Finally, just touching a little bit on helminths, which are multicellular. They actually have tissue systems, like a digestive system in some cases. So they are animals, but we are lumping them in with our study of microorganisms because they are associated with infectious diseases and have microscopic stages of their life cycle. They do not have a cell wall. They are all chemoheterotrophs and have some sort of either absorptive or ingestive through they have these kind of suckers that will latch onto the tissue wherever they, often in your gut, because you're exposed sometimes through contaminated food, and can have very complex life cycles, both outside of humans as a host, have an intermediate host, for example, like snails or other organisms, and then have us as humans as a host. So complex life cycles that can sometimes be human pathogens. Finally, getting to the microbes, the microbial agents, the non-living agents that are part of this that are members of this microbial world that I'm introducing you to. They are all, by definition, acellular. That means they lack a cell structure. They have no cell membrane. They may have a lipid component to them in the case of some viruses that are enveloped, but they all lack those internal organelles, no nucleus, no mitochondrion, no ER that is necessary for them to reproduce independently. So because they can't reproduce independently, by definition, current definition, some virologists may beg to differ, but by current definition of microbiologists as well as biologists, they are non-living. So we call them microbial agents, microbes. They have characteristics of life. They may have genetic material. Viruses either have DNA or RNA, never both. Viroids are simply RNA, and prions are simply a protein. In the case of viruses, they're a little more complicated in that they also have a protective protein coat that is around them in all cases. And some have, in addition to that protein coat around the nuclear material, the nucleic acid, have an envelope of a lipid bilayer. And we'll talk more about that important distinction in their anatomy. So here's an example of a virus here colored in the scanning electron micrograph. That's a human T cell, a T lymphocyte, a white blood cell that's being infected by HIV, the virus. So the red or orange is the HIV, and the green is the host cell. And so you can see here the relative size of these non-living agents, of microbial agents. They're much, much smaller, and they're simpler by definition. So no cell structure, DNA or RNA in the case of HIV. It's a retrovirus, has RNA that's converted back into DNA as part of its life cycle, has a lipid envelope, so a lipid bilayer around it in addition to the protein coat in the case of HIV. Not all viruses have that, but HIV does. And it replicates only, not outside here as we see it, but inside, because it requires, it has to hijack our own cells to use the energy in those cells and the machinery, the ribosomes that allow it to make protein from that genetic information that it carries. And then finally, as I mentioned before, viroids are plant viruses. They cause plant diseases. They have no protein coat, just RNA. They usually get into plants by a break in the tissue of the leaf or somewhere in the plant and just transfer from cell to cell once they get in that tissue. And prions are these really wacky infectious proteins that are, unlike any other protein, very resistant to denaturation or messing up the shape of the protein by heat. So heat will not, our normal methods of sterilizing and protecting us from infection don't work well with prions because they're resistant not only to heat but to enzymes that normally degrade proteins. So really challenging in the medical community to deal with, and mistakes have been made. It's a very slow progressing disease, but by eating meat that has that's come from a cow with this neurodegenerative disease called mad cow disease, you can acquire a neurodegenerative illness. Or if you eat if you consume human tissues that are from a person who already has prion disease, or genetically you can inherit the misfolded disease form of the infectious protein, and eventually later in your life acquire Creutzfeldt-Jakob syndrome, which is a progressive neurodegenerative infection.